We are recording. Turn the light off. Do you have any questions before we get started? Hopefully more people show up. It is a Monday though. Anyway, even though it's not written on the board or on the side, I'll remind you, and anybody watching the video, you can still retake exam 1 for the practice. You cannot retake it to change grade. You can still retake exam 2. Retake it as many times as you want until it closes, which will be when exam 3 is closed, and I don't know when that's going to be yet. For exam 2, again, even if you've already contacted me and I've already adjusted your grade, it's not too late. You can still, looking at the wrong side here. Anyway, it's not too late. You can still continue to retake exam 2. Of course, exam 1, you can meet online and get all the correct answers and 5% boost the grade. Exam 2, you'll be able to do that once it closes for retake. That's it. So any questions about retakes? It's not written on the board, but today, or this week's lab is 100% virtual. So just like last week, if you happen to get it done in time and I happen to get it graded in time, I might tell you don't even need to come in. But other than that, you should come in for lab this week. Again, it is 100% virtual, so we will all be doing our own thing, but I'll be here to help you. So any questions about that? Okay, what else? Yeah, I think that's it. It's the usual announcements and reminders. I don't have anything new to say. So you guys have anything new to say? Any announcements of your own or reminders or questions? All right then. In that case, it's not quite 8 yet. That's all right. There's nothing new here, so I'll start with this too. The normal attendance stuff. I have to turn in attendance records to the university, so it's mandated for that reason. But also, as you know, 100 points of your grade this semester, which is an entire letter grade, is for your participation points. You get those different ways, and we're going to do it the same way we've been doing it. I'll probably change it up eventually again, just to make sure you guys are on your toes, because that's the purpose of it, is to make sure you're paying attention. But today is like it usually is, so you're going to write down the attendance words as I say them. If you're watching the video, you're going to take a screenshot every time I do that. So you can send an email that includes all those attendance words, and also one question about the material covered in the lecture. But try to make it about the material covered in the lecture. If you happen to have a question like, hey, when's the next exam? Include that too if you want. But not for points. For points, ask a question about the material. I have one person who takes 101-01. He's not even in this class. And he always sends, when he does this for extra credit, he'll send in those words or the emails. And he always has like test questions that I can use. Like here's a question that I wrote that could be used on an exam or a test. That's good because it reinforces that he knows that material. And also it's helpful for me because sometimes I'm like, yeah, that is a good question. I can use it. But also, I think maybe even more importantly, sometimes he'll write a question and he also provides the answers to his questions. And he'll say, and this is the correct answer. Sometimes it's wrong, and I can use that as a learning moment. So anyway, the point is, ask a question about the material. For the participation points, the question should be about the material. But obviously, you can go beyond that and ask me any questions you want. But for the points, ask a question about the material covered. So for example, if I say something like, hey, my kids are on spring break this week, so I will not be in early every day this week, which is true. Then at the end, don't ask me a question like, where do your kids go to school? Well, you can, but not for the points. Anyway, any questions? Okay, so we'll get started. The first word for attendance will be video. I will now take attendance. Then we'll get started. Ashley is here. Barrett is here. Wooten is not here. Jury is not here. Dickens not here. Clark is not here. Inca is not here. Isaiah is not here. Smith is not here. Watson's not here. Kaylee's not here. Lucas is not here. Marley is not here. Noah is here. Savannah is not here. Anita is here. Edwards is here. And the jury just came in. Did I miss anybody? Okay, let's get back into it then. Hope you guys had a good weekend. Speaking of weekends, I did not do any work over the weekend. So if you send me any emails or turn anything in, I have a lot of catching up to do. So it might be a while before I get to it. I just apologize in advance. But that is why. Anyway, here's where we left off. You learned about how genetics works, right? You learned about dominant versus recessive and things like that. We took a long time talking about that. And then eventually on the last lecture, I said, wait a minute, but that's not actually how things work. So here's the exceptions to everything you've learned. And then we talked about codominance, and you're going to need to know codominance because, again, like I said on Friday, for the exam, I'm going to give you some example, like this thing made it with this thing, and these were the results. And from those results, you need to understand or figure out if it was an example of codominance, where the phenotypes of both parents show up, or if it was an example of incomplete dominance, where both of the phenotypes of the parents blended together, or if it was a case of complete dominance, which is what we learned prior to that, which is just the simple one's dominant over the other. And that's where we left off. So now we're talking about some other exceptions to the rules. So are there any questions about all that, about the recap? All right, the next one's called pleiotropy. And in my opinion, this is a very simple concept. The hardest thing I would guess is just remembering that pleiotropy means this, right? Because the name doesn't really explain it, right? Codominance, that kind of explains the situation. Incomplete dominance, that kind of explains the situation. Complete dominance, that kind of explains the situation. Pleiotropy, unless you're really good with your Latin, probably doesn't make sense. But let's talk about it. Pleiotropy is a situation where we have one gene, which is what we've been studying. We always study one gene, one character, right? But now we're talking about one gene that affects several characters. So that's the first time we've talked about this. Again, everything else we've always talked about has been one gene, one character, right? One gene for blood type, for example, even though there was 3 different alleles, which was also a new thing at the time for us. One gene for chin dimple, one gene for eye cover. But in all actuality, there are other instances, and this is called pleiotropy. And one example is sickle cell disease. I'm not going to ask you any questions about this. Here we go, I'll talk about it anyway. And you're going to see, we're going to talk about sickle cell disease a lot in different contexts here towards the end of the semester. Obviously, we're talking about it right now as an example for pleiotropy. Later, we're going to talk about it as an example for mutations. We already talked about mutations previously, but once we really start talking about molecular biology, we're going to talk about it again, and that will be the example. And then finally, we're going to talk about it again in natural selection. So you're going to hear a lot about sickle cell disease, but anyway, sickle cell disease is a great example of pleiotropy, right? Because this is one gene, but it causes all these different symptoms. And in these cases, these symptoms are characters. And the way it works is when you have a normal gene, your hemoglobin, right? You make that protein normally, so you have a normal shaped hemoglobin. Therefore, you have a normal shaped red blood cell. Therefore, you don't have any of these issues. But with this situation, there's one mutation, right? One letter is off. So the protein is shaped wrong. Therefore, the cell that you can see over there is shaped incorrectly. And because of that, you have all these different symptoms. Like, I guess I didn't have them written down. Doesn't matter. No, the next slide I'll show you has the symptoms. Not that it matters though, because again, sickle cell is just a disease, or excuse me, just an example. So usually it's only the homozygotes that have the disease. So for example, if you were a carrier, if you're heterozygote, you probably don't have the disease, but the book did say usually, so obviously there are some exceptions to those rules. So any questions? Well, I won't take questions yet. The next slide is a visualization of this. Does anybody need me to wait? Okay, there it is. So again, if somebody's homozygous for this allele, then they have this abnormal hemoglobin, therefore they have the abnormal shaped blood cell, and then you can see here these symptoms, right? Weakness, pain, organ damage, paralysis. Just an example. I was actually watching a show called The Pit. I've heard a lot of good things about it. I finally started watching it over the weekend. And they had somebody who came in with sickle cell disease. I thought, hey, I teach about that. Anyway, any questions about pleiotropy? That's it for pleiotropy. Again, one gene, many characters. And also, again, to remind you how the exam is going to go, I'm going to make up probably some hypothetical fake thing that's not real, so it's not going to help you to memorize all real-life examples of pleiotropy, but I'll make something up, and if it's this one gene, and then it describes more than one character, you should say, oh yeah, that's pleiotropy. Where it gets a little bit more confusing, in my opinion, is the next one. These two things by themselves aren't that confusing. Pleiotropy that we just talked about, not that confusing, right? But the next one, that's misspelled. The next one should be called polygenic inheritance. And that's a little bit more complicated because, again, I don't know, for me, polygenic inheritance, it kind of sounds like what we just talked about. Polygenes. So don't let that get confusing. But in this case, it's kind of like the opposite of pleiotropy. So even though the name can be confusing, if you think hard about the name, it does kind of differentiate between the last one. Polygenic, as in many genes. Think about it that way. So now we're not talking about one gene with many characters. We're talking about many genes with one character. Or as the textbook says, the additive effect of two or more genes on a single phenotype. Again, in a sense, this is the opposite of what we just talked about. It's the opposite of pleiotropy. So this is going to be a different situation for the test. It would be really hard to get an example of this. These things made it, here was the results. So the question about this is your main bullet point right here, as far as the exam is concerned. If you were to graph, basically, if you were to graph the phenotypes, right, if you were to do a Punnett square, and I'll show you an example on the next slide. If you were to do a Punnett square and graph the different probabilities, then you would get a bell curve. But think about that. If you were to graph, it wouldn't look like that if you were to do that for chin dimple, for example, right? And you guys did something like this at the end of that one lab, right? So if you just did the number of people that have chin dimples in the world, I don't know what the number would be, but let's just say it might be like that, right? You would have two bars, right? You either have a chin dimple or you don't. So when you graph the phenotypes for chin dimple, you have a 2-bar graph. If you were to graph something for polygenic inheritance, because you would have all these different phenotypes, you would have a bell curve. And that's exactly what the question is going to hint at on the exam. Evidence points to height as being an example of this, according to the textbook, but in all actuality, it is. Height probably does have a number of genes that affect it, but also there's other things like nutrition. So I don't know if that's the best example, but it's a good example because everybody's familiar with it. And that goes back to this example too. If you were to graph the height of everybody on Earth, that would be a lot different, right? You wouldn't have 2 bars, but you could have as many bars as you want, right? Because it all depends on which bar represents, right? You can do it by centimeters. So everybody who's 150 centimeters, 151 centimeters, 152, right? If you were to graph it, it would probably look something like this. You would have a lot more of the people that are average and a lot fewer of the people who are on the extremes. And that's what this next slide shows. And one of you, I think I remember who, it doesn't matter, but one of you asked a great question about Punnett squares. Like, how big can a Punnett square get? And the answer is as big as you want it to get. We have about 20,000 genes. So technically you could do a Punnett square for 20,000 genes, but that's beside the point. But here's the Punnett square. So far, the Punnett squares that you've done were monohybrid crosses, right? You're only looking at one character. Then I showed you some examples of dihybrid crosses. So you had 2 characters. Remember, that was a pretty big box. That was a large box. Now we're looking at 3 genes, right? So three genes, 1 character. But look at this Punnett square. So it gets a lot larger. But again, yes, when you graph the phenotypes, you get a bell curve. You don't get 2 bars like you would for something like chin dimple. Again, you either have it or don't. You get a bell curve. That being said, I want to point out, as complicated as this looks, right? And you can see these new complicated ratios, 1 to 64, 6 to 64, 15 to 64, 20 to 54, or 64, so on and so forth, right? Even as complicated as this looks, this is actually an oversimplification. Height is not quite this simple, but it's a good example. And they did, like I said, they just took it one step farther than what you've seen before. So it was a trihybrid cross instead of a dihybrid cross. Anyway, any questions about that? All right, so again, remember, for polygenic inheritance, it's many genes for one character, and the way you would identify it and the way the question is going to be written is going to have something to do with a bell curve, natural distribution. All right, there's no question about that. Here's the last example of the variations on Mendel's law. This is epigenetics, and this a little bit goes towards what I was saying with height, but not exactly. Let's talk about it. So many characters are a result of genes and the environment. And that's basically what epigenetics is. Basically, as far as we're concerned, that's what epigenetics is. It's where we're not just talking about your genes, we're also talking about the environment. And before I move forward too, I'm going to use an analogy that I'll use later when we really start talking about how this stuff works. Right now, we're just scratching the surface. Right now, we're talking about inheritance, like we're just looking at the ratios, right? The patterns of inheritance. Later, we're going to get into molecular biology and talk about how this works at the molecular level. So, addressing this one bullet point, let me give you an analogy that I'm going to use later when we start talking about that. your chromosomes have genes on them, right? So you have 23 pairs of chromosomes. So you can almost think of it like you have 23 pairs of instructions, like books that have instructions on how to build proteins. That's what genes are, right? Instructions on how to build proteins. So you might have genes that say, this is how you build this thing. But just like any book, sometimes it's hard to get to that particular page. Right? Maybe the page is stuck or crumbled. And also, just like any book, sometimes it's really easy to get to a page. Maybe somebody left the book upside down, so now it just like naturally flops over to that page, right? Or maybe there's stops, or maybe there's a smudge on the page, so it's harder to read. That's kind of like how you can think of genes in the environment. You have this set of genes, it's instructions on how to build proteins, but sometimes the environment affects how well you can read those set of instructions. Right? Sometimes it boosts it, sometimes it makes it harder to read, but the environment plays a factor, and that's exactly what happens when we are genes. Anyway, whether characters are more influenced by genes or environment is a very old and hotly contested debate, right? Nature versus nurture. There's still a lot of things that people talk about with that, especially these days. You know, people might talk about, you know, especially gender, the idea of gender. Anyway, this is not that kind of class, so you can look into those things if you want. The book does point out, though, if you spend a lot of time with identical twins, it is very convincing that the environment, not just the genes, that affect a person's trait. Because again, you know, identical twins have identical genes, right? They have the same exact genome. But like the book was saying, if you spend enough time with a pair of identical twins, you start to notice their differences. And that would have to be because of the environment, right? They have the same genes, so the environment does play a role. Anyway, as far as the exam is concerned, I mean, really, this slide is pretty unimportant. This is more like the introduction to what we're talking about. If anything, you just need to know, at least from this bullet point, that first bullet point, right? Basically, we're saying, when we're talking about epigenetics, you should think genes and the environment and how they affect each other, or how the environment affects the gene. So any questions so far? Okay, so in general, it's only the genetic influences that are inherited, right? So in general, this is what we've thought for the longest time, where it's only your genes, right? So, for example, if you don't use sunscreen, and you get a lot of UV energy, and it mutates your DNA, technically, that can be passed along to your offspring. Of course, you have to be made nude, but that's a whole different thing. Anyway, the point is that, you know, we know it's genetic things that are passed along. We've always known, well, not always, for the longest time, that's what we've known. And we've always thought that the effects of the environment are not passed down to the next generation. Meaning, like, you know, if you have healthy genes for lungs, but you're a smoker, you're going to ruin your lungs with, you know, environmental things. But it's not like when you have a kid, the kid's going to come out with, you know, smoker's cough, right? Because that is not what's passed along. However, in recent years, and I guess it's not that recent anymore because this particular textbook was written a while ago, biologists have begun to recognize the importance of epigenetic inheritance. So this is relatively new. I'm probably not going to ask any questions about it. Just let me know about it. And epigenetic inheritance is the transmission of traits by mechanisms not directly involving the DNA sequence. So remember, your DNA is just a bunch of letters, A's, C's, T's, and G's. That's the instructions. The instructions to build these proteins are written in that language. So what we're saying is we knew that if you messed up that language, if you mutated it, that could get passed along, right? That could be inherited. But what we're saying here is now we're starting to understand that there's other mechanisms. Like you could keep the same DNA sequence, but still change how your genes are read, basically. And sometimes that can be passed along. Right? That's basically as deep as we're going to get into it. And I'll give you an example on the next slide. But yeah, so again, the point is, I don't want to get too deep into it, but so remember when we talked about what a chromosome is, right? We said it's that DNA and it's wrapped around those histones, and we call each one of those little balls of histones, those proteins, we call them nucleosomes, right? So it's around pretty tightly around there. But what you haven't learned yet is there's that machinery that clamps onto your DNA and reads the gene, and that's how we express our genes. So sometimes it's just harder to read those genes because of the things like histones and other things that you haven't learned yet. There's other components to the DNA that can affect how the DNA is read, right? So the sequence may stay the same, Something might happen to the chromosome that makes it harder or easier to read that particular DNA sequence. So here's an example. Chromosomes can be chemically modified by adding or removing chemical groups to the DNA. And again, that's what you haven't even learned about that. You haven't learned about chemical groups. So far, the only thing you know is the chromosomes are DNA, about 50% DNA and about 50% protein. But remember, proteins are made up of those 20 different amino acids. Each amino acid has its own side chain. So obviously there's a lot of room for adjusting it, right? So we can adjust these proteins, not even the DNA. So again, the DNA remains the same when we're talking about epigenetics. DNA remains the same, but we do some things chemically to it to where, again, some genes are harder to read, some genes are easier. So the environment plays a role in making these changes. And this also may explain how one identical twin can suffer from a genetically based disease where the other twin doesn't. So that does happen, right? So we would have two identical twins that have the same exact genome, but only one of them has this genetic disease. The other one still has the genes for the genetic disease. They just don't have the disease because it hasn't been triggered by the environment to chemically alter the DNA, not the sequence, but the DNA itself. The chromosome itself. So simply put, epigenetics changes the DNA/chromosome, right, the structure, while genetics is strictly the sequence of the letters, right, the A's, the T's, the C's, and the G's. Any questions about this slide? It's really long-winded for us. It's a pretty simple concept, which is that the environment also plays a role. All this, and really that's all I'm still saying as far as the exam is concerned. The environment does affect how we express our genes. Very good? All right, so epigenetic modifications and the changes in the gene activity that result from those modifications may be carried on to the next generation. So that's kind of what we were getting at there too. Not that that would be a test question. Again, as far as the exam is concerned, we're already done. You know what you need to know, which is the environment. It also plays a role, and that's called epigenetics. But yes, just for your own knowledge, we are learning now that these epigenetic modifications can be transmitted to the next generation. So that's the bad news. The good news is that second bullet point. Like once you change the sequence of your DNA, once you do there's a mutation. As far as now, anyway, that's it, right? The mutation's there. But unlike that, epigenetic modifications can be reversed. So that is the good news. And for extra credit, if you want to look into this for a paper, look up some examples. What are some real life examples of how someone can epigenetically alter their genome? Like what particular things? Like, is it smoking, sunbathing? Is it snorting asbestos? Whatever it may be, right? Look up some real life examples of how this works. But anyway, are there any questions? We're now done talking about epigenetics, and we are now done talking about the variations on Mendel's law. Meaning, once we're done with this slide, we're going on to the last of the three major bullet points of this chapter. All right, so if there's no questions, the next word for attendance is going to be role, R-O-L-E. Role is the second word for attendance. And even though I didn't mention it this morning, I'm going to say it out loud to remind you and to remind myself. I'm still giving you extra credit. So again, if I give out 5 attendance words and you send in 4, that's full credit. If I can give out 5 attendance words and you send in all 5, that's extra credit. Of course, that's only for those of you who are here physically. If you're watching the video or you're online, that doesn't count. Anyway, again, we're finally to the last of the 3 major bullet points of this chapter, which is the chromosomal basis of inheritance. Good news is we can go through this pretty quickly because the way your textbook teaches it, it taught you all the stuff I taught you, but it didn't really talk about it from the point of view of the chromosomes. But as I was teaching it, I did. So everything I'm about to teach, most of the things I'm about to teach you, not all of it, but most of it I've already covered, just not officially. So again, as I was explaining, independent assortment, I was explaining how the chromosomes work. The textbook wasn't, but now that they're done talking about all this, they're like bringing it back. They're rewinding it and say, all right, let's talk about this again, but let's really focus on the chromosomes. So let's jump into it. Might be an early day, we'll see. Anyway, the chromosomal theory of inheritance states the genes are located at specific positions on chromosomes. Like I said, No new information here, right? You already knew that. So we have gene locus, right? That's plural. Loci, singular. You already knew that. So, and I always use the example, or just use some random example, like if the gene for crooked teeth is on the bottom of chromosome 13 for me, that means for every other human on Earth that doesn't have a chromosomal abnormality, the gene for crooked teeth will be found at the same exact location on the same exact chromosome. So granted, they might have a different version of it, right? That might be an allele, but that's where the gene will be found. So it's the behavior of the chromosomes during meiosis and fertilization that accounts for the inheritance patterns. So again, I've already said this too. As I was teaching you, these inheritance patterns, like these, when you cross two purebreds, then like homozygous dominant and homozygous recessive, then the result will always be a heterozygote, right? When I taught you that, I was talking about the chromosomes, so this is nothing new for you either. And this too, this is bullet point two. This is just a reminder of everything you've already learned. It's the chromosomes that undergo segregation, right? It's the chromosomes that undergo independent assortment. Therefore, it's the chromosomes that account for all these Mendel's laws that you've recently learned in this chapter. So in a sense, this slide is more like a reminder of what you already learned and an introduction to what you're about to learn, but there's no real new information on here. It's just taking different information that you learned throughout the chapter and put it all on one slide. And I'm not necessarily going to ask you any questions like, there will be no test question when I say, well, what does the chromosome theory of inheritance state, right? I'm not going to ask you that. I just want you to understand, again, all these things about Mendel's law that you learned, I just want you to understand it's because it's chromosomes. As a matter of fact, some of you, when you sent in your attendance words, asked questions about these things, and I explained it from the point of view of the chromosome. I was explaining how it works at the chromosome. Here's some examples. We've already talked about, this is our first example of independent assortment, when we were talking about, instead of looking at one trait, we're looking at two traits. So in this case, we're looking at the P color, yellow being dominant and green being recessive. But we're also looking at the shape of the P, so smooth versus wrinkled. Smooth being dominant and wrinkled being recessive. And this is what you've already seen. You've already seen this picture in the previous slides, except now they drew little chromosomes on there too to show you, like, okay, when we say that this thing is big Y, big Y, big R, and this thing is little r, little y, little y, instead of just showing you that like it shows right here, we also show you the actual chromosomes, right? So this thing goes through meiosis. Those two big Ys separate from each other. Those R's separate from each other. Therefore, the gamete is only left with one of each. But because it's homozygous dominant for both, you have a homozygous dominant there, dominant there, and a dominant there, right? The dominant on that chromosome for the R gene, dominant on that other chromosome for the Y gene, and then of course the opposite over there. And then they come together and you have a heterozygote, right? So again, nothing new on this slide, except now the old textbook is just showing it to you, but also with the picture of the chromosomes. So you can see how it's these chromosomes, right? These chromosomes that get lined up in the middle of the cell and then separate, right? And then the sister chromatids lining up in the middle of the cell and then separating. So it's a great thing to study if you want to. It's a good visualization, but this is not one of those things where it's best for me to sit here and talk to you about it. This is one of those things that's best to look at it and understand the picture. So this picture's not in the textbook. You can download the PowerPoint if you want to look at it. But again, this is everything you already know about, meiosis and independent assortment and segregation. That now they're showing you visually with the pictures of the chromosomes. And there's the bigger picture. So it's really complicated. Well, not complicated. It's just a lot of information on the slide. All right. So any questions about that? Okay. So that was just the introduction to these three things we're going to talk about. We're going to talk about linked genes, which we've hinted at before. We're going to talk about sex determination, which we've already talked about. And then we're going to talk about sex-linked genes, which I'm kind of hinting at too. So let's jump into it. Linked genes. These are genes that are located near each other on the same chromosome. They are linked. So this is sort of like, I don't want to say an exception, but in a way, it's like an exception to the rule of what you just learned about independent assortment. Right? So to remind you, when we're talking about independent assortment, what we're saying is that genes don't travel together, right? So and I think it was Inca, she's not here, but I think she asked me a question in the emails. And I think the example I gave was a real life example. And I don't remember the chromosome numbers, but there's a gene responsible for eye color that's on one chromosome. There's a gene that's responsible for hair color, or whether or not you have red hair. That's a better way to put it. And that's located on another chromosome. And my point was, in that example that I gave her, because those are on different chromosomes, they don't travel together, right? So, for example, just because you got your mom's eyes doesn't mean you're going to get her hair. You could, but you might get that chromosome from your mom for your eyes, but then you get the chromosome from your dad for your hair, right? So they don't necessarily travel together because they're on completely different chromosomes. So when those chromosomes line up in the middle of the cell during meiosis one, it's not like all mom chromosomes are on one side and dad's chromosomes on the other, right? It's all random. This is an exception to that because in this case, they are on the same gene. Excuse me, all these genes are on the same chromosome. Therefore, and again, I'm just making this up. Let's say gene 1 is for bad breath and gene 2 is for back hair, right? So let's say your mom's got hairy back and bad breath. And this is her chromosome, right? So if she's got bad breath and you got her gene for bad breath, you very well likely may have got her gene for hairy back as well, because it's on the same chromosome and you got her chromosome. So let me ask you this, since it's not on there. Why would that not happen though, right? Like if you got your mom's bad breath gene, then sometimes, again, most of the times you do get all these genes because they're on the same chromosome, but sometimes it doesn't happen that way. So why do you think it would happen? What do you think would cause that to happen? Where you would get your mom's bad breath gene, but somehow got your dad's gene too, your dad's hairy back gene. How would that happen? Because it's not on the side. What would cause that? You have to think about some of the stuff we talked about. I'll give you a hint. Prophase 1. So the first prophase of meiosis. It's something that happens there that you need to know for the exam. Regardless of linked genes, you need to know this thing happens in prophase 1. All right, I'll give you another hint with my horrible drawing skills. So this is a homologous chromosome, right? I don't know what chromosome number that is, but whoops. That's so bad. Let's say this is chromosome number 3, whatever. And there's the one from your mom, there's the one from your dad. So again, let's say you got your mom's gene 1, but somehow you got your dad's gene 2, your dad's gene 3, and your dad's gene 4. And if we were to change, do the opposite, right? This is your dad's chromosome, so that would be dad's gene 1, mom's gene 2, mom's gene 3, and mom's gene 4. How in the world would that happen? How can you have a chromosome that has dad chromosomes up here, but mom chromosomes down there? How can that happen? No guesses? Hopefully you're just being shy. Last chance for a little bit of extra credit. Anyone want to guess? What is it?
Does it split off?
Split off, yes, good. And there's a word for that, and this is what you guys need to know. Crossing over. Remember? When the chromosomes, so the homologous chromosomes get close together, sometimes there's crossing over, meaning they're swapping chunks. So you would have a mom chromosome with some dad chunks on it, and a dad chromosome with some mom chunks on it. So that's the answer. But anyway, all that being said, this is, and when you send in your words, remind me that you said that, so you get a little extra credit. Anyway, what happens is, when we're looking at two different characters, right, a dihybrid cross, what you should remember too, this is another thing you should recognize, that 9 to 3 to 3 to 1 ratio, the genotype ratio, right? So, well, you've seen it before. Anyway, 9 to 3 to 3 to 1 ratio is the ratio we would expect when two genes are completely not related to each other because they're on different chromosomes. What we're saying here, and this is your book may get deeper into it, I don't remember, but I don't, I'm leaving it at this. What I'm saying here is, if you see some other 4-number ratio that's not 9 to 3 to 3 to 1, you should think to yourself, okay, that's an example of linked genes, because these genes must have been on the same chromosome because they're traveling together. Anyway, another thing that your book might talk about, I'm not going to get this deep into the exam. Hopefully common sense would tell you this, but more than likely, these two genes are probably more likely to be inherited together than, for example, gene 4 versus gene 1. And if we had more time, I would ask you, why is that? But the answer, again, is crossing over, right? So remember, crossing over always happens. Sometimes there's a small chunk, sometimes there's a big chunk, sometimes it's a medium chunk, sometimes it doesn't happen at all. The point is, if you have crossing over like right there, for example, obviously these genes are going to travel together. These genes are really far apart. Imagine gene up here too, that's even farther apart. So then, the farther apart they are, the less likely they are to travel together, thanks to linked genes. So that's it for linked genes. Any questions about that? All right, this one's pretty simple too. Sex determination in humans. And I want to be very clear, this has been very controversial recently. We're talking about sex. I'm not talking about gender. Gender is a social construct. Sex is a little bit more straightforward. What I'm about to teach you for this 100 level non-biology class is very straightforward. But in all honesty, it goes a little bit deeper than what I'm about to teach you. So let's talk about it. Many animals, including mammals, have a pair of sex chromosomes designated X and Y that determine the sex. Before I move forward, I'll point this out too. This is a good opportunity for extra credit if you want to look into it. What are some of the exceptions to this? Like, again, I said including mammals. So what about other animals? What about flies? What about snakes? What about coral or sponges? There's a lot of different animals other than mammals. So if you want to look into it, like what is the sex determination for other mammals or other animals? And for that matter, why even look at animals? Broaden it even more. Like, plants have sex. They have a male and a female. For example, pepper plant, when you look at that flower, that is both male and female parts. You're looking at a male and a female all together. Anyway, as far as we're concerned, males have one X chromosome and one Y chromosome. Females have two X chromosomes. So for the exam, that's what you need to know. And you already know from previous discussions that that's not always the case either. Even though, right, so sometimes you might have a male who has an extra Y chromosome, or a male who has two extra Y chromosomes, right? So X, Y, Y, Y. Sometimes you'll have a female that has an extra X chromosome, or a missing X chromosome. And this is just the chromosomes, again, and that's where we're leaving it for this 100 level class. But again, I just want to let you know, it gets deeper than that, right? So there's these genes on these chromosomes that are making a male, and a female, a female. But just like any other genes, there could be something wrong. There could be mutations, there could be extra copies of the genes, fewer copies of the genes, epigenetic problems, so where some of these genes are always turned on, or some of them can never be turned on, right? So again, it gets more complicated than what I'm teaching you. This is 100 level course. These are the basics. All right, any questions about this slide? Here's a picture of the sex chromosomes for a male. You can see the X chromosome over there, Y chromosome over there. That Y chromosome is a very tiny, tiny chromosome, smallest in our genome. So that being said, let's talk about sex-linked genes. That's the last discussion for this chapter. Let's see. You know what? That's not the next word for tense. I know I circled it, but that's not the next word for tense. Let's move forward. So a gene that is located on a sex chromosome is a sex-linked gene. Who'd have thunk it, right? A gene that's located on a sex chromosome is called a sex-linked gene. Here's an easy question for you, and I guess we do have the time, so I will sit and wait for the answer. For extra credit, most sex-linked genes are found on the X chromosome, not the Y. Why do you think that is? Don't overthink it. If I were to ask a kindergartner and show them this picture, say, hey, look, there's an X chromosome, there's a Y chromosome. Why are there more genes on the X chromosome? Because they wouldn't be overthinking it. They would just look at it and come up with the answer right away. Noah's already answered one. Anybody else want to guess? Why are there more genes on the X chromosome and not the Y? Yes, ma'am.
It's bigger.
It's bigger. Thank you. Yes, that's it, right? It's a lot. It's literally just genes. It's DNA, and the instructions are written in the DNA. So yeah, it's bigger. Remind me that you answered that, please, when you send in your attendance numbers. All right, so I'm going to give you some examples of actual sex-linked genes, some conditions like red-green color blindness, hemophilia, a particular type of muscular dystrophy. These are all sex-linked recessive alleles. What you don't need to know, though, that's just an example. So I'll put it next to this to indicate when you're studying, don't feel like I'm going to ask you, what are some examples of some sex-linked genes? I'm probably not going to ask you that. I do want you to understand how it works. Also, I want to point out, again, to reiterate what's on here, what we're talking about is, in this particular example anyway, are recessive conditions, right? And notice, when I first started this chapter, I don't know if anybody noticed, but I used to say to have the recessive condition you would have to be homozygous recessive, or I would say both of your alleles have to be recessive. Then, starting at least in the previous discussion, instead of saying that, then I changed my verbiage a little bit, and I used to say, or I started saying, to have the recessive condition, 100% of your alleles have to be recessive. And it might sound like I'm saying the same thing, but I'm not. 100% of your alleles being recessive is not the same thing as saying both of your alleles being recessive. And that's what we're getting at here. Because with sex-linked genes, and this will make a lot more sense in the next few slides when I show you Punnett square, if it's on the X chromosome, and you're a male, then you only have one allele, right? Because you only have a male only has one X chromosome. So if you have that one recessive allele and you're a male, that means 100% of your alleles are recessive, right? So that would be a more accurate thing to say than both of your alleles being recessive, because you're a male, therefore you only have one X chromosome, therefore you only have one of those alleles, therefore you're not going to have two alleles. So that's why I'm starting to say 100% of your alleles need to be recessive to have the recessive condition. Anyway, let's jump into this example. Again, I'm not going to ask you any questions about it. It's just an example. Irregular color blindness is a common sex-linked disorder caused by a malfunction of light-sensitive cells in the eyes, and it's mostly the males that are affected, which should tell you, or before I even jump into that, sorry, heterozygous females seem to have some defects, but not too much. And the next slide shows a test. But anyway, the point here is, hopefully you can determine, and you might get a question like this, if I were to give you this information on the exam and say, which chromosome is this on, the X or the Y? You should be able to figure out from that information that it's on the X chromosome. First of all, the fact that it's mostly males that are affected, because again, if it's on the X chromosome, and if it's a male, then all you need is that one allele, then you're going to have the recessive condition. So there's your first red flag. The other part is when we're talking about heterozygous females, obviously, if it's a heterozygous female, they don't even have a Y chromosome, right? So we know you should be able to deduce that is on the X chromosome. So you might get a question like that on the exam, where I give you some random thing like this and say, hey, which sex chromosome is that on? So any questions about that? All right, can anybody, let's see if I can see it. It's a lot more clear on the screen, but I can see it. Can anybody not tell me, does anybody not know what number that is? Just out of curiosity. You can't see it?
Yeah, I'll learn it.
Okay, so you do have it, okay. Anybody else? It's a 7, but yeah. I had to go through this test to join the military, because my job included things with colors. Actually, I don't know if everybody has to do that or not. Anyway, because they're located on sex chromosomes, sex-linked genes exhibit unusual inheritance patterns, which I was getting at earlier, right? You should be able to deduce from what I said that was on the X chromosome. But I'm going to give you some examples, some other examples. We're going to look at some Punnett squares. So we're looking at some different possibilities. And again, we're going to use red-green color blindness as an example. And again, everything I'm about to teach you is just an example. So don't feel like when you're studying, you need to memorize any of this. These are just some examples so you can look at how it works. All right, so we're still dealing with a Punnett square. But you can see these are written a little bit differently. So we have a female here, X and an X, and we have a male, an X and a Y. And since this gene is just represented with an N, and it's only on the X chromosome, so you can see here, this female is homozygous dominant. That male, even though there's only one allele, because he's a male, is homozygous recessive, because 100% of his alleles are recessive. Anyway, when this person goes through meiosis, half of their sperm are going to have that X with a little N, half of the sperm are going to have a Y. When this female goes through meiosis, half of her eggs are going to be, 100% of her eggs are going to be dominant, right? Because either one, she's homozygous dominant. But anyway, those are the results. Then when you cross them, and again, you might get a question like this on the exam, or I ask you to set up a Punnett square and say, what are the chances of them having a female who is completely normal? And that would be 0%, right? I'll get to that in a second. What if I said, what's the chances of having a female that's a carrier? You can see both of those are carriers, so I would say 50% chance of having a female that's a carrier. What are the chances of having a male that's normal? And that's 100%, right? And what are the chances of having a male that is colorblind? 0%. So any questions about that? All right. Let's move forward then. Give you another example. This time it's a carrier female mating with a non-affected male. And remember, last time we had a homozygous dominant female and a homozygous recessive female. And then again, you could just do the Punnett square. And again, I could give you questions. What are the chances of having a completely normal female? 25%. What are the chances of having a female who's a carrier? 25%. What are the chances of having a completely normal male? 25%. And what are the chances of having a colorblind male? 25%. So again, these are just examples. These are some questions you might get on the exam. One last example. Here we have a carrier female mating with a colorblind male. And then again, if you do the Punnett square, what are the chances of having a completely normal female? 0%. What are the chances of having a completely normal male? 25%. What are the chances of having a carrier female? 25%. What are the chances of having a colorblind female? 25%. And what are the chances of having a colorblind male? 25%. Or if I really want to get, I can ask more questions. What are the chances of having a colorblind kid? Forget male or female. Then you could look and say, oh, it's 50%. Anyway, any questions about that last example? And once again, this is just an example. I can ask you specific questions about this. For the exam, I'm going to ask you to do at least a couple Punnett squares. What are the chances this thing mates with this thing? What are the chances of the offspring being that? And some of you have done it, but to remind you, there's an extra credit worksheet out there that does nothing but give you practice with Punnett squares. You can do that for practice, but also for extra credit. Extra credit linked to your genetics lab, by the way. So however many points you earn on that extra credit worksheet be applied directly to your genetics lab. And if there's any points left over, that will be applied to your course total. Anyway, here's another example. Nothing to write down here. This is just an example. I'm not even sure this is from this new textbook. I know it's from the old textbook. But hemophilia is a great example. You don't need to know any of this. This is just another example. One of the things I like about this example is if you're into history, then that's I don't know, this is one of those interesting situations where biology and history overlap. And I don't know, I mean, I like history, but I'm not an expert. And also, this is very specific. But if I remember correctly, because this kid had hemophilia, they had this guy, Rasputin, who was there living with the family, with the royal family, to help, with this kid who had hemophilia. But he was also a really weird character, and he was also really influential to the royal family. And because of that, basically, there was a lot of mistrust with the people of Russia. So eventually they had a revolution. And part of that was because of this guy who was living with the royal family, because their kid had hemophilia. So without the hemophilia, you may have never had that Russian Revolution. At least that's the way I remember it. Again, I'm not claiming to be an expert. But it's another good extra credit opportunity if you want to write a paper about that. Look into it. What are some times when history has been influenced by biology? Especially back in the day. Like I just started recently watching a documentary from Ken Burns about the American Revolution. A lot of it does have to do with disease, right? So disease definitely changed the course of history, especially back in the day. And that's biology. Anyway, leaving that, you can read that if you want. Once again, this PowerPoint was based off the old textbook. The old textbook at the end of the chapter always had this thing called evolution connection, where they talk about evolution in the context of whatever that chapter is about. So once again, I'll never test you on that, but I included it there if you wanted to, if you want to look into it. The next word for attendance is wolf, right there. And again, you can look at that if you want. Really quickly, I'm going to show you some other stuff. This is stuff that's on the slide. I think this will help you study, but this is not on your old textbook. So if you look at this stuff and you find it helpful to understand how things work, then download your PowerPoint and study it. But yeah, it's a summary of the key concepts, right? The law of segregation. So I would say that's one of the biggest. I get more questions about that and independent assortment than anything else when I ask you guys to ask a question about the material. So really quickly, just to make sure you understand before we move forward. Law of segregation, basically, as far as the exam is concerned, what we're focused on here is that you have two alleles. Generally, we're going to oversimplify because sometimes that's not the case. Sometimes it's more complicated, as you've learned. But generally speaking, you have two alleles for each gene, right? They might be the same version. There might be different versions, whatever. You have two alleles. You got one from your mom, one from your dad. When you go through meiosis, those alleles get segregated, so the resulting gamete, right, is only going to have one allele. That's the key thing you need to focus on when it comes to the exam. When we're talking about segregation, the alleles are segregated. Therefore, your gametes only have one of the alleles, because you're supposed to have two alleles for each gene. But your gametes only have one, because they segregated. Any questions about that? All right, this one's pretty simple. I do like this picture though, because this kind of goes back to what I was saying earlier. So I used to have these pictures. You see pictures like this, similar to this, where it's like, all right, that thing crossed with this thing, and then it would show just one flower. But I like this because there's more flowers. But in this particular case, they're trying to make a point, right? So they're saying this is like a test cross, right? So you know this thing's dominant because it's purple, but you don't know its genotype, right? You don't know if it's big P or big P or big P little p. You know, because that's a white flower, that it has to be homozygous recessive. So you want to find out the genotype of that thing. You cross these and you look at the offspring. If you get some purples and some whites, then you know for a fact this thing had to be heterozygous. If you get 100% dominant, purple, you don't know for a fact, but it strongly points to the fact that this must be homozygous dominant. And to remind you what I said back when we were talking about this with the Labrador retrievers, that other example. Remember, just because they're all purple does not necessarily mean it's definitely PP, big P, big P. It's just most likely, right? Because the chances of them, a big P, the chances of a heterozygote crossing with a homozygous recessive and getting 100% purple, it's a very low probability, but not impossible. Right? So it is technically possible to cross this and get that, but it's not possible to cross this and get that, right? If that's homozygous dominant, there's no way you would have a white flower. Anyway, study that if you want really quickly. That's an example of incomplete dominance, right? So we have a blend of the two genotypes. That's a good visualization of pleiotropy, right? You have one gene, multiple characters. Here's the opposite, right? Polygenic inheritance, multiple genes, one trait. Again, males are X and Y, females are X and X. Here's a little table for sex-linked traits. Normal female, carrier female, blah, blah, blah. Everybody said you can study that. Some other examples, and the last word for attendance will be offspring. And that's it. We are, oh wow, I thought we were done early, but we were done right on time. So yeah, that's it. I'll be in my office and online for office hours. And again, I have catching up to do. So if you need me in a hurry, call my office. If you email me, it'll be a while because I have catching up.